So, when you're looking at acute versus chronic versus latent, remember acute, short term, and it's short term because the pathogen is eliminated, right? And a lot of times with acute infections, you can become immune to reinfection. When you have chronic illnesses, they last for long periods of time. With latency, the illness can actually recur over time. Now, in terms of where the pathogen is, they can be localized or systemic. With localized infections, the microbe might be limited to a small area. For example, boils caused by Staph aureus. In a systemic infection, the agent will actually spread throughout your entire body, something like the measles. Now, you can have words dealing with where the pathogen is. So whenever you hear emia, you should just assume it has something to do with the blood. So bacteremia, you have bacteria circulating in the blood. This does not actually mean you have a disease state. You brush your teeth and you have bacteremia. Now, toxemia deals with toxins circulating through the bloodstream. Viremia means you have viruses. Sepsis is when you actually have an acute life-threatening inflammation caused by agents in the bloodstream. Now, remember, we have Koch's postulates, right? He was the guy that said you have to actually pull the pathogen from an organism, and every organism you're pulling it from, it has to look the same when you grow it. You then purify it, and you put it in a healthy individual. That healthy individual should get the disease, and when you reculture the organism from that newly infected host, it should look like what you started with, right? However, remember, we have limitations with this. Not everything can be grown in the lab. Some individuals that have the disease will not always show symptoms. Some things actually have multiple microbes causing the disease, and we don't always have good animal hosts for testing. This is why we've had to switch to a more molecular approach for Koch's postulates, where you're looking for virulence factor genes or certain products being formed. That way, you say that the certain molecular sequence is always there. Now, how do organisms become pathogenic? So some can actually produce toxins that you ingest. Others will grow on various mucous membranes. Some can actually invade host tissues and avoid your immune system. Others will invade host tissues and produce toxins. And you basically have this evolutionary arms race where pathogens and hosts are trying to evolve towards a balanced pathogenicity. Now, how do you establish an infection? Well, you have to adhere to the host cell. So adhesions will attach to host cell receptors, and these adhesions are oftentimes associated with fimbriae. They tend to be very highly specific to exploit certain cells. You can have colonization with growth in biofilms. You can have avoidance of secretory IgA, and you're going to find that these organisms will compete with normal microbiota and can sometimes start tolerating the toxins they produce. You can also have pathogens delivering effector proteins to host cells. So gram-negatives can actually inject molecules other than proteins into cells to kill them or damage them. So you do have to get through biological barriers as a pathogen. So the skin is actually really hard to penetrate. So many bacteria that colonize the skin actually rely on wound injuries to get in. Staph aureus will enter through a cut or a wound. Yersinia pestis will get past the skin because it's injected through a flea bite. You can also have bacteria penetrating mucous membranes. This is an entry point for a lot of pathogens. They can be directly uptaken by cells. And what happens is the pathogen, the bacteria, actually tricks the cell to engulf it using endocytosis so that it can get inside of the cell to start causing disease. So you can also have exploitation of antigens. So some pathogens will actually trick M cells, allowing them to get across the intestinal barrier. You even have some invasions by alveolar macrophages where they basically trick the macrophage to take them up, but then the macrophage never activates to actually destroy them. Some bacteria can even hide within a host cell. So this allows them to avoid proteins that might target them, phagocytation, and antibodies. So Shigella can actually direct transfer from intestinal epithelial cells to adjacent cells by causing host cell actin polymerization. Listeria does something similar. Some bacteria can also avoid destruction by phagocytes, so they find ways to avoid them, either by avoiding recognition or preventing attachment. So some bacteria can be serum resistant. Neisseria can actually hijack the host system and bind certain proteins to avoid the attack complexes from recognizing them. Others can avoid the antibodies, so they will actually mimic molecules on the host cell, and they basically will take those recognition proteins, copy them, and then put those copies on themselves to appear as self. Now, damage can be caused to the host through direct means, through toxins produced, but you can also have indirect damage. Many diseases cause damage because of the immune response to the pathogen, not the pathogen itself. And when you have damage, it can actually help the pathogen to exit the body and spread elsewhere. When you look at Vibrio, it causes a very violent watery diarrhea. You can have up to 20 liters of diarrhea per day. Think about that. That is 10 2-liter bottles of soda. That is going to contaminate any water supply it comes near. Pertussis actually triggers this violent coughing. That's going to release the pathogens in the air for other people to inhale them. So exotoxins are going to be proteins that are produced with specific damaging effects. They are either secreted or leak into the tissue when the bacterial cell breaks. This can be a problem. Some bacteria, it is not enough to kill them if they are causing an infection. You also have to deal with the toxins. Now, these toxins can act locally or systemically. Vaccines are critical, so toxoids are going to be inactivated toxins found in vaccines. So antitoxins are suspensions of neutralizing antibodies. Neurotoxins will specifically damage the nervous system. Enterotoxins will damage the intestinal system, usually by causing some form of diarrhea. Cytotoxins can actually damage a variety of cell types. So here are some exotoxins produced by primary pathogens. Now, you can also have damage caused by inflammation. So these products being produced as part of the inflammatory response can actually damage the host. So there's also damage associated with adaptive immunity. Immune complexes that target the bacteria can actually settle and form, especially in places like the kidneys and joints. This can lead to inflammation. You can also have cross-reactive antibodies, so instead of targeting the bacteria, they make a mistake and they target the body's own tissues, creating an autoimmune response. Now, in order to infect a host, a virus has to be able to enter the appropriate cell, hijack the host machinery, avoid recognition and destruction by the host, and move on to infect new hosts. So damage to the host can actually come directly from the viral infection or because of immune responses to the virus. So with viruses, they can bind to host cells as part of the invasion process because they're looking, well, they're not looking because they're not alive. They're targeting specific receptors. They can avoid the antiviral effects of interferons, so they can actually block the expression of certain host genes and enzymes that target viruses. And viruses can avoid antibodies by moving from cell to cell or causing syncytia to form. Remember, syncytia are those giant multinucleated cells. They can also modify the surface antigens and basically out-evolve the body in terms of its capacity to produce effective antibodies. And then they can take advantage of antibodies to facilitate macrophage uptake. So some viruses can also avoid immune responses because they can interfere with apoptosis, So remember, P53 helps cause apoptosis, which is programmed cell death. And if certain cells have a virus inside of them, they will begin apoptosis. But certain viruses can actually prevent that from taking place so that the cell doesn't self-destruct despite being damaged. Now, when you look at eukaryotes that cause disease, with fungi, most of them are saprophytes. So remember, they absorb their food, and the ones that cause disease are usually opportunistic. Many of them are actually dermatophytes, so they will cause superficial infections on the outside of the body, places like the hair, skin, and nails. You can have more serious fungal infections. Typically, though, you have to be immune compromised. If you have dimorphic fungi, they actually, remember, will change shape depending on their environment. So some can actually be molds in the environment and then develop into yeasts internally. Again, your immune system will usually clear these out unless you're immune compromised, and then you have a problem. The other issue is that fungi can produce mycotoxins, and those can be toxic. And you have allergic reactions to the cnida, which are the reproductive spores of fungi, and this can also, aside from allergies, this can trigger asthma. When we look at protozoans and helminths, many of them actually live within the intestinal tract, or they're going to enter through the bite of an arthropod vector. They will attach to certain host cells looking for specific receptors, and they have different mechanisms to avoid antibodies. So Plasmodium, which remember causes malaria, it will actually produce enzymes to allow it to enter red blood cells, and once it's in the red blood cell, it's kind of good to go. When you look at the trypanosomes, they will actually vary their surface antigens to keep tricking the immune system. And the damage caused by eukaryotic parasites really depends. They can compete for nutrients with the host. They can actually block the intestines. And depending on what enzymes are being produced, you can have large immune responses. All right, that is what we have for chapter 16.